So thank you for the introduction, and yes, we're gonna steamroll through some of the work I did during my PhD, looking at identifying a novel genetic suppressor for Barth syndrome. Okay, so Barth syndrome is considered a rare X-linked gene with a prevalence about 1 in 300,000 patients, but this might be highly underdiagnosed just due to the pleiotropy of the disease. So there's documented mutations across the entire open reading frame of TAS, and the patients present cardiovascular phenotypes, skeletal muscle phenotypes, central nervous system phenotypes, metabolic phenotypes, even immune phenotypes such as neutropenia. And both the age of onset, the severity, and which symptoms arise and what combinations are quite different across patients, and there's currently no targeted therapeutics available. Once diagnosed, essentially you're trying to get them a new heart. But this is a case where we can see that there are probably genetic modifiers at play. So the root of this is mutations in the gene TAS, and TAS encodes an acyltransferase which remodels the signature phospholipid of the mitochondria, being cardiolipin. And it's critical for cristae formation, holding the electron transport chain in place, as well as apoptotic mechanisms. And even though it's such a critical enzyme in cardiolipin biosynthesis with no known redundancy, and being highly conserved, there are still huge gaps in our knowledge as to why perturbation of TAS manifests the way it does and results in Barth syndrome. But the hallmark feature is an elevated ratio of an intermediate form of cardiolipin called monolysocardiolipin to mature cardiolipin. So the way we're going about this is, because this is a classic genotype to phenotype problem, and a case of missing heritability, just with the complexity of the disease despite one known gene being responsible, is we're approaching it by looking at genetic interactions. So how does a gene influence the effect of another? And in the most simplistic case, genetic interactions occur when mutations in 2 or more genes combine to produce an unexpected phenotype. So we can look at gene A and gene B. If you knock out either, the cells can still be viable, but if you knock out both gene A and B, you can have a negative genetic interaction, with the extreme case being synthetic lethality, and then essentially you have all your cells have died, or a positive genetic interaction where you have some type of growth advantage. And this can be a rare case of genetic suppression. So our lab does this by profiling genome-wide CRISPR screens in human cell lines, and we use the HAP1 cell line as a model, and we use a CRISPR screening approach to do this. So we use the genome-wide TKOV3 guide RNA library, and we essentially infect our cells, either being parental cells or what we call a query, where we've got a mutation of interest, and that you can think of as your gene A, and we want to look at all possible combinations of gene B across the genome. And then we're going to profile guide RNA abundance by next-generation sequencing. So we'll look at our reference time point and our terminal time point, and then we've got bioinformatic approaches on how we can quantitatively decipher a genetic interaction. So I applied this pipeline to uncover the genetic interactions of TAS. So using 5 independent CRISPR screens and different clones, different media conditions, I was able to produce this robust genetic interaction profile. So everything in yellow are positive genetic interactions on the scatter plot, and everything in blue are negative genetic interactions. And I focused on the positive interactions because I want to see, can we alleviate the phenotypes of Barth syndrome at all? So I focused on this gene ABHD18, which at the time was only known as C4ORF29. So I did a validation, I made clonal cell lines, and indeed, the double knockout grew to wild-type level, so it totally restored the fitness effect of a TAS cell line. And then I also screened ABHD18 and found that it had reciprocal genetic interactions, with TAS being its top positive, as well as shared synthetic lethality, suggesting that they may be working together. But since we knew nothing about ABHD18, the first thing was I needed to localize where is the protein. So I tagged it with M-neon and overexpressed it, and essentially found that it co-localized quite well with the mitochondria. I also then performed bio-ID experiments in 2 different media conditions, glucose and galactose, to favor mitochondrial condition, and found that a number of the interactors were within the intermitochondrial membrane space, specifically interacting with components of the electron transport chain, as well as a number of transporters and other metabolic lipid mitochondrial genes. So kind of supporting the localization of ABHD18 being mitochondrial, and then we kind of came up with this idea that it might be in the intermembrane space similar to tafazin. So importantly, I told you that the hallmark feature of Barth syndrome is an elevated cardiolipin or monolysocardiolipin to cardiolipin ratio. When we knock out ABHD18 in a TAS mutant background, we can actually restore that MLCL to CL ratio. And what was interesting is that we found ABHD18 on its own had very little monolysocardiolipin being generated. So the dot plots are just showing you species of both cardiolipin and monolysocardiolipin based on double bond, as well as cardiolipin. We saw more nascent forms of cardiolipin and not mature forms of cardiolipin being produced with ABHD18, suggesting that ABHD18 might function in the cardiolipin biosynthesis pathway, specifically acting on monolysocardiolipin. And indeed, that's the case. We directly assessed this by doing thin layer chromatography with our collaborators at Scenic Biotech in Amsterdam. And we found that despite which cardiolipin substrate you use, if we have wild-type ABHD18, we could produce monolysocardiolipin, but we also, so ABHD18 being an alpha-beta hydrolase fold domain-containing protein, we found that there was active site serine that we predicted to be the catalytic site. We mutated that to serine to alanine, so that S199A mutant, we can see that we're not producing monolysocardiolipin anymore. So then we can see an AlphaFold rendition of what the protein looks like. We did a GROMACS molecular docking model of taking a monolysocardiolipin molecule, and indeed it actually went into this pocket of ABHD18 with the S199 catalytic site. So we think we found the missing lipase in human cells, generating monolysocardiolipin to cardiolipin that's previously kind of been elusive. So next, we wanted to test this in mice. So again, with Scenic in Amsterdam, we generated an ABHD18 knockout mouse as well as a double knockout mouse, and we restored both Barth human phenotypes as well as all the phenotypes we see in mice. So we fixed the Mendelian birth ratios in males, we fixed the weight, we restored fertility, so male mice and Barth patients are infertile. We can now see sperm in the lumen in the double knockouts. We also restored cardiac functioning that we observed with fractional shortening. So at 10 weeks of age, we did echocardiograms on these mice, as well as looked at a number of cardiac stress markers that are often profiled in Barth syndrome patients, and again found that the double mutant restored these to near wild type. We also restored the cardiolipin, monolysocardiolipin ratios and saw the same thing that we were seeing in cell lines. And in doing transcriptomics of the mice heart, we saw a huge amount of dysregulated genes in TAS mutant mice, but that we've restored these and essentially rescued near all of these deregulated genes in the double knockout. So we also, with another industry collaborator at Lundbeck, generated a small molecule covalent inhibitor for ABHD18 that we're calling ABD646. And with Seema Mattel's lab, we got Barth patient fibroblast cells, treated these cells, and within 96 hours, we can see a restoration of fitness effects. Doing TEM, we can see mitochondrial morphology being restored. And then with Ian's lab, we also tried to look at zebrafish embryos with TAS morpholinos, and this is probably the most striking result, that in a TAS morpholino treated fish embryo, we can see this kind of catastrophic cardiac phenotype, but in the double or with treatment of ABD646, we see restoration of both the morphology. I don't think this video is going to play, but we also would be able to see it in terms of contraction. So they're essentially near wild-type hearts again with 3 days of treatment in the zebrafish embryos. So my last slide is, right now there's no targeted therapeutics for Barth syndrome. The foundation is pushing the FDA to approve this drug called elamipretide that's known to bind to cardiolipin. I tested elamipretide, and you can see despite increasing doses, we saw essentially no effect in increasing the fitness in TAS cells, but in comparison to our ABD646 treatment, with only 5 days of treatment, we saw significant rescue and fitness. So what we're working on now is just doing these further drug testing on our mouse models, and hopefully we've uncovered the first targeted therapeutic for Barth syndrome and other cardiolipin-related disorders. So there's been a lot of people involved in the project, so I'm thanking them there, and thank you very much.